Hello, hi, Assalamualaikum dan selamat sejahtera to all my lovely students. Alright, so for today, we are going to learn last chapter in your syllabus. Okay, what is the chapter? It is chapter 13, Genetic Technology. So in this chapter, we are going to learn about the technology, about the utilization of the technology in the genetic field. Alright, so let's take a look on the learning standards that you have to cover in this subtopic. Alright, so we are going to learn 13.1 genetic engineering. Right, genetic engineering. So what are the learning standards that you are going to cover for today? Okay, number one, describe the terms of genetic engineering. Genetic engineering. And the second one, genetically modified organisms or GMO. Alright, so genetically modified organism, in other words, is GMO. Okay, genetically modified organisms. Okay, second learning standard, explain the application of genetic engineering in the production of genetically modified organisms. Okay, and then genetically modified foods. And last one is insulin. Alright, so these are referred to the learning standard that you have to understand for today. Okay, let's take a look on the terms of the genetic engineering and genetically modified organism. So what is a genetic engineering? Genetic engineering. So it's a gene manipulation technique to modify. To modify what? To modify an organism genetic material to produce the new combination of genes. Alright, so the keywords for the genetic engineering is a gene manipulation technique. Gene manipulation technique. Right? Teknik yang mengubah suai gene, right? To modify an organism's genetic material to produce new combination of genes. Okay, that is referred to the genetic engineering. Right, so what is a genetically modified organism? So an organism such as animal, plants or microorganism. Okay, microorganism that is produced by recombinant DNA technology is known as genetically modified organism or GMO. Alright, so this is referred to the genetically modified organism or GMO. So what is a GMO? Refer to the an organism. That refer to the animal, plant or microorganism that is produced by the recombinant DNA technology. Recombinant DNA technology. Okay, later I will explain about the recombinant DNA technology. Alright, so make sure you can know the terms of the genetic engineering and also the genetically modified organism. Alright, so genetic engineering involves the transfer of a segment of a DNA segment from one organism to another organism. Alright, okay, by DNA recombinant technology. Okay, by DNA recombinant technology. So genetic engineering will involve the transfer of a DNA segment from one organism to another organism. Okay, by what technology? By what technique? By DNA recombinant technology. Okay, so by using the techniques and procedures in the recombinant technology, so biologists can recombine the DNA or genome. So genome. What is referred to the genome? Genome refers to the complete set of DNA of an organism. Okay, so actually what is a DNA recombinant technology? It is a technology used to transfer the DNA segment from one organism to another organism. And at the end of the techniques, so the biologists can recombine the DNA or the genome of an organism. So at the end of the technique, yeah, it will produce the new combination of a gene, of the gene of an organism. All right. So that is referred to the DNA recombinant technology. It is a technology that transfers the DNA segment from one organism to another organism. And at the end of the technique, at the end of the process, uh, it will produce the new combination of the DNA or the genome of an organism. Alright, so that is referred to the genetic engineering, and then the DNA recombinant technology. Alright, you can refer the genome definition in your textbook on page 280 in bioexploration. So please go there and read the definition of the genome. Alright, so that is referred to the genetic engineering and genetically modified organisms. Okay, so genetically modified organisms are organisms that contain recombinant DNA. Right? Genetically modified organisms are referred to the organisms that contain recombinant DNA. Right? Okay, so the recombinant DNA technology enables the production of new gene combination. Recombinant DNA technology enables the production of new gene combination. So an organism that contains recombinant DNA is known as a transgenic organism. It is referred to the terms, a new terms for you, refer to the transgenic organism. Refer to the organism that contains recombinant DNA. Or you can say that they are GMO, genetically modified organisms. The example of the genetically modified organisms is cow. Cow. Okay, so this genetically modified cow produce milk which does not have or does not contain beta-lactoglobulin, a type of protein that causes allergy among some children. So now the cow is genetically modified. So when the gene is modified, so this cow will produce milk that doesn't have any beta-lactoglobulin. So what is a beta-lactoglobulin? It is a type of protein that causes allergy in some children. Alright, so this is one of the example of the GMO, genetically modified organism. Okay, the other example is goat, right? Okay, goat. So this genetically modified goat possess or has the human gene which codes for blood clotting factors. Right, okay, so this goat has the gene, has the human gene which code for blood clotting factors. So this blood coagulation factor is found in the goat's milk and can be purified and used to treat hemophilic patients. Okay, do you still remember what is a hemophilic patient? So they are referred to a person that have hemophilia. hemophilia. Okay, what is a hemophilia? It is referred to the disease that cause excessive bleeding or non-stop bleeding. All right. So this goat will produce milk. That milk contain the coagulation factors or the blood clotting factors that can be purified, and then it will be used to treat the hemophilic patient. Alright, now we go to the genetically modified food. Genetically modified food. Okay. So just now we are talking about the genetically modified organism. Okay, now we go to the genetically modified food. Okay, focus to the food. Okay, recombinant DNA technology has successfully produced many beneficial varieties of crops. Okay, so such as paddy, oil palm, pineapple, corn, soya bean, and also the livestock. Livestock. Okay, so crops merujuk kepada hasil tanaman. Livestock refer to the ternakan. For example, salmon, cattle, and goat. So genetically modified food possess or has DNA from other species of plant or animals. Consumption of the GMF by humans may cause health implications which are still unknown. Alright, okay, so these are referred to the genetically modified food. Modified food that refer to the varieties of the crops and also the varieties of the livestock. Alright. Alright, so let's go to the characteristics of genetically modified food. The characteristics of GMF. Alright, so they are resistance to herbicides. Okay, what is a herbicide? Herbicide refer to the chemical or refer to the substance that kill the plants, especially weeds. Especially weeds. So it will kill the unwanted plants, such as weeds. Okay, and then resistant to disease, right? Resistant to the disease, and then application in the medicine. Application in the medicine. Tolerance to heavy metals such as plumber, mercury, and so on, right? And then resistance to pests. Alright, so these are referred to the characteristics of the GMF. Resistance to herbicide, resistant to the disease. Okay, so it will not get easily infected with the disease. And application in the medicine, used in the medicine. Tolerance to heavy metals and also resistant to the pests. Alright, so there are the characteristics of the genetically modified food. Okay, now let's go to the advantages and disadvantages of the GMF, genetically modified food. Okay, what are the advantages? Okay, overcome worldwide food shortage. Overcome worldwide food shortage by producing high quality transgenic crops. So it will produce a high quality transgenic crops. Okay, what is a transgenic? Transgenic refer to the organisms that contain the recombinant DNA and also producing high quality of the livestock. Okay, and then second one, reduce cost of food production. Reduce cost of food production because the food is produced in large quantities and also in very high speed. And the second one, increase the nutritional value. Okay, the third one, increase nutritional values of the crops. Okay, so it will produce maybe a bigger fruit, right? Okay, and then number 4, reduce probable problems of crops related to pests. Okay, so because most of the GMF are tolerant to the pests or resistant to the pests. reduce usage of the pesticides, right? Okay. Then last one, increase in production, reduces price of food, thus increasing food availability. Okay, so these are referred to the advantages of the GMF. Okay, we go to the disadvantages. Yeah, what are the disadvantages of the GMF? Endangered natural species. Okay, so because of the transgenic organism, transgenic organism refer to the organism that have recombinant DNA. So means it has a new combination of the genes. So it will endanger the natural species. Yeah? So there is a slight possibility that the foreign gene in GMF may be transferred to humans, for example, antibiotic resistant genes. So this is still unknown, right? They are still do the study. So may have adverse effect on human health and genetic material. So this is also still in study. Right? So we don't know they are maybe have the adverse effect in our health and also in our genetic material. Okay, so let's go to the example of the GMF, genetically modified food. Okay, example is in the corn production, ya, dalam penghasilan jagung, penghasilan jagung di sini. Right, so a Bacillus thuringiensis. Yeah, a Bacillus thuringiensis. So what is a Bacillus thuringiensis? It is one of the bacteria. Yeah. So this Bacillus thuringiensis gene, yeah, the gene of this bacteria is inserted into corn to increase its resistance against insect pests. Okay, so this is referred to the one example of the GMF. GMF involved here is the corn. Yeah, corn. So in the corn production, okay, what happened? A gene from the Bacillus thuringiensis is inserted into the corn, yeah, DNA, into the corn DNA, and then it will produce the corn, yeah, that is resistant to the pests. Okay, so another example of the genetically modified food is super salmon. Yes, super salmon. Okay, as you can see from the diagram here. So the salmon here, this one is bigger than this one. Right, okay. So what is a super salmon? It is a genetically modified fish which is approved by the Food and Drug Administration as a safe food for consumption in USA, United States of America and Canada. So it is modified by inserting a growth hormone gene, a growth hormone gene from a Chinook salmon into the genome of an Atlantic salmon. So hence, a super salmon is created which grows at a faster rate and can be produced throughout the year. Alright, so the super salmon is produced when when the growth hormone gene from a Chinook salmon is inserted into the gene of the Atlantic salmon. So it will produce the combination of the gene and the salmon will become a super big, super bigger. Alright, okay, bigger than the original salmon. Okay, so this is another example of the GMF, genetically modified food. Okay, the other example is potato. Okay, potato is an important crop to people who live in cold climates. In cold climates. So it is very sensitive to low temperature and frost. Okay, so the potato is easily to frost. Okay, a gene from Arabidopsis, Arabidopsis species plant, which enables the plant to tolerate freezing condition. Okay. So now the plant is tolerate the freezing condition. Okay, because the gene from the Arabidopsis is inserted into the potato genome, to create a genetically modified potato that can grow well in cold climates. Okay, so this is referred to the example of the GMF, genetically modified food, which is referred to the potato. So the potato is the GMF potato now can grow well in cold climates. Okay, so I hope you can understand here the GMF or the genetically modified organisms is produced when the DNA recombinant technology, the DNA recombinant technology are utilized to produce the new combination of the genes, and the new combination of the genes will produce the genetically modified organisms. Okay. Alright, so now let's go to the insulin. Insulin. So what is insulin? Insulin is a type of hormone. Hormone. So insulin is an important hormone which controls blood glucose level. Okay, do you still remember what is the function of the insulin? Insulin will convert the excess glucose, into the glycogen, into the glycogen. Okay, in the older days, right, insulin was extracted from the pancreas of cattle or pigs to treat diabetes mellitus patient. Right, so that is in the older days, But in nowadays, it is no more extracted from the cattle or pigs. Okay, so nowadays insulin can be commercially produced by genetic engineering. For patients with diabetes mellitus. Okay, beside insulin, other success of genetic engineering include hepatitis B vaccine, blood clotting factor, and also growth hormone. Okay, so that is referred to the insulin. Insulin refer to the hormones that convert excess glucose into the glycogen. So in the diabetes mellitus patient, there is a excessive glucose in the blood. So they have to get the injection of the insulin. Because their pancreas cannot produce enough insulin, okay, in the diabetes mellitus patient. Okay, so how to get the insulin? So nowadays the insulin can be commercially produced by genetic engineering. Right? Okay, so let's go to the production of insulin by genetic engineering technique. Okay, how does the insulin is produced in the genetic engineering technique or DNA recombinant technology? Okay, okay, so this is referred to the splice of the human DNA with insulin gene. Insulin gene is here in the yellow color. Insulin gene. So the insulin gene is cut with the restriction enzymes. The insulin gene is cut with the restriction enzymes. And then here, okay, so this is referred to the plasmid. Okay, this is referred to the plasmid. Okay, what is a plasmid? A plasmid is a circular DNA found in bacteria. In bacteria, which is used as a cloning vector. So, this technique, in the genetic engineering technique, in the production of the insulin, the bacteria is used here. The plasmid of the bacteria is used here as a cloning vector. So the plasmid is cut. The plasmid is cut, right, with a restriction enzyme. So this is referred to the cut plasmid. Okay, and then what happened next? Yeah, okay, next. So they are recombinant. Yeah, they are combined. They are combined. The insulin gene of the human here combined with the cut plasmid here. Okay, by DNA ligase enzymes. Okay, by DNA ligase enzyme and produce a recombinant plasmid. Produce a recombinant plasmid. Okay, what is a recombinant plasmid? It is referred to the recombinant plasmid which is successfully inserted with human insulin gene. Yeah, it is referred to the plasmid that is inserted with the human insulin gene. Okay, as you can see here, human insulin gene is inserted into a plasmid. Okay, and then what happened? The recombinant plasmid is introduced into a bacterium. So this is referred to the bacterium. Yeah, bacterium. Okay, so they are introduced. So the recombinant plasmid here introduced into a bacterium. Okay, so after the recombinant plasmid is introduced into a bacterium here, so now the bacterium now is called as a transgenic bacterium. Okay, why transgenic bacterium? Because this bacteria contain recombinant DNA or recombinant plasmid here. Yeah, it has a recombinant plasmid here or recombinant DNA. And okay, we go to the number 5. Bacterial clone multiplies. Okay, so the bacteria will multiply so many times and will produce insulin. And then number 6, insulin is extracted from the transgenic bacteria and purified. Okay, so that's how the insulin is produced. Okay, is produced in genetic engineering. So they will use the bacteria, yeah, the plasmid of the bacteria and the bacteria itself, yeah, to produce the insulin. So now the bacteria will produce the insulin hormone, and then the insulin will be extracted, yeah, extracted from the bacteria and will be purified, and it will commercially used, yeah, to treat the for the using of the diabetes mellitus patient. Alright, I think that's all for now. So make sure yeah, you able to describe the terms of the genetic engineering, genetically modified organisms, and also explain the application of the genetic engineering in the GMO, GMF, and also in the production of the insulin. Okay, please read again and again and try to understand the concept of the genetic engineering. Alright, because this is referred to the technology, yeah, refer to the technology that you are going to learn more, yeah, in the future. Alright, so I think that's all for now. So take care everyone, stay safe.